Roots. Two main patterns of root growth: taproot system and fibrous root system. Taproot system has a large main root known as taproot that function to tap deep source of water. Taproot develops directly from radicle and produces lateral roots. Lateral roots develop their own branch roots, resulting in an excessive root system. Taproots generally penetrate deeply and are therefore well suited for plants becoming larger each year. Fibrous root system, usually in seedless vascular plants and most monocots such as grasses. No single root stops. Adventitious roots. Instead of one main root developing from the radicle soon dies and numerous roots arises from the lower part of the stem. These are called the adventitious roots. Form lateral roots, typically shallower and more horizontal than a taproot system. Quickly obtain water.
Roots has three overlapping regions: inner root, division, growth, differentiation of cells can be traced linearly through three overlapping regions. First is the zone of cell division. It consists of the root apical meristem and three primary meristem. Second is the zone of elongation. Overlaps with zone of cell division, where the derivatives stop dividing and begin to grow in length. Lastly, the zone of maturation overlaps with zone of elongation, where the cells begin specializing in structure and function into different cell types such as epidermal cells and conducting cells. Quiescent center. The initial root apical meristem located within small spherical center of meristem. Root hairs. It is primarily within the zone of maturation, beyond the zone of elongation, that a root produces the epidermal cells. Root hairs specialize to absorb water and minerals from the soil. As the roots continue to grow, older hair die and new one arise in each zone of maturation. Cross-section of a root. Stele, the central cylinder of a root or stem. Protostele, simplest type of the stele. Cortex, solid central cylinder surround the protostele. Root has two important cell layers. Pericycle, give rise to lateral root or branch root. Endodermis, regulates the flow of substances between cortex and vascular tissue. For further knowledge about mycorrhizae, mycorrhizae is a mutualistic association between vascular plant root and soil fungi. Mycorrhizae has two main types, the endomycorrhizae and the ectomycorrhizae. The endomycorrhizae, fungi penetrate plant root and produce branching structure called arbuscle. And the ectomycorrhizae, fungi do not penetrate, instead surround the roots to produce sheath called mantle.
Leaves originate on the shoot apical meristem as bumps of tissue.
As the apical meristem grows up and away, the cells in the leaf buttress elongate until flattened leaf primordia appears. Leaf epidermis, a single layer of cells derived from phytoderm, protects the leaf from water loss, abrasions, and the entry of fungi and bacteria that cause disease. It regulates the exchange of gases such as CO2 and O2. and water vapors that are needed by or produced by the leaf.
In studying the plants, including the leaves, we should have to study also the tips, margins, veins, shapes, and arrangements of each leaves of plants.
Leaf veins. Seeds know there are typically one or more small vascular bundles called leaf trays, which leave the main vascular system of the stem and pass through a connecting petiole to reach the leaf. Leaf shapes and arrangements. Larger leaves with pin. Smooth blades occur in environments with polar temperature, lower levels of light, moist conditions, and no wind. Smaller leaves with variable margins occur in the environments with warmer temperatures, high level of light. Leaf epidermis. A single layer of cells derived from phytoderm protects the leaf from water loss, abrasions, and the entry of fungi and bacteria that cause disease. It regulates the exchange of gases such as CO2 and O2 and water vapors that are needed by or produced by the leaf.
The major parts of a plant, the stems, describes the shoot apical meristem as again divided into three regions: the central mother cell zone, peripheral zone, and the pith zone. The central mother cell zone, it contains cells that divided and frequently and give rise to the cells of the peripheral and pith zone. The peripheral zone are the three-dimensional rings surrounding the central mother zone, consist of cells that divided rapidly to become leaf primordial and parts of the stem, supplying cells to protoderm, procambium, and other parts of the ground meristem that produces cortex. Pith zone is below the central and peripheral zone, which produces cells that become the part of the ground meristem that produces pith. Cell layer model, also called the tonical corpus model, describe the initial of the shoot apical meristem as forming several cell layers, starting from the tip of apical meristem